You know that feeling, right? You're sitting in an exam, you're sure you knew the answer, but when you get the results back, the marks just aren't there. Well, we're going to fix that. Today, we're going to dive deep into a real GCSE biology question, and I mean deep, using the official mark scheme to see exactly how to bag every single point. So how do you do it? The secret is to switch hats. You've got to stop thinking like a student and start thinking like the person with the red pen, the examiner. We're going to figure out what exactly they're looking for to award those precious marks. All right, let's get right into it. We've got question 01 from a real GCSE biology paper, and it's all about milk decay. And the best part? We're going to use the official mark scheme as our guide. This means you'll see the exact phrases and keywords that get you full credit. You ready for this? Okay, so here's the plan. We'll kick off with the milk decay question itself, talk about microorganisms and pH, then move on to designing the experiment. After that, it's all about interpreting the results, explaining the science behind it, and then nailing that final answer. Let's go.
All right, section 1, the milk decay question. This is all about the basics of decay. So first things first, the question's going to test you on the fundamental stuff. Who are the main culprits when it comes to decay?
So here it is. Question 01.1. Name 2 types of microorganism that cause decay. Nice and simple, for 2 marks. This should be a really quick win if you've got your revision down. So, what does the mark scheme want to see?
And there you have it, straight from the horse's mouth. The mark scheme says bacteria and fungi. Simple as that. But wait, check out the extra info. They'll allow mold, makes sense, it's a type of fungus. But look at this, do not accept viruses. Now why is that? It's a great question. See, viruses are parasites. They need a living host to replicate. They don't actually eat or break down dead stuff like bacteria and fungi do. And that little detail, that precision, is exactly what we're talking about.
Okay, let's keep building on this. Next up, we're looking at how this biological process, the decay, actually leads to a chemical change, something you can measure. All right, question 01.2. As milk decays, lipids are broken down, and it asks you to name the product that causes the pH to decrease. So you've got a massive clue right there. Lipids are breaking down and the pH is dropping. What does a drop in pH mean? It's getting more acidic. So what's making it acidic? Just a 1 marker? And the answer is fatty acids. It's right there in the name, isn't it? When those microorganisms break down lipids, which are just fats, they create fatty acids. And what are acids going to do? They're going to drop the pH. That's a perfect, straightforward answer for 1 easy mark. Okay, time to switch gears a bit. We're moving away from just pure knowledge recall and into the realm of scientific skills. This next bit is all about how you'd actually set up a proper, fair test to investigate this stuff. So another 1 marker coming up. This one's about your practical knowledge. How would you go about measuring that pH change we were just discussing? The mark scheme gives you a couple of options. Universal indicator, either the paper or the solution, or a pH meter. Both are perfect, but here's the trap. Look at that note. Just writing pH paper won't get you the mark. Why? Because the examiner wants universal indicator. That's the one that gives you a specific color for a specific pH value. Something like litmus paper is way too basic. It just tells you acid or alkali, not how acidic. See, it's these tiny details that can make or break your answer. Next up, 2 marks on the table for this one. Give 2 control variables the student should have used. Remember what control variables are, right? They're all the things you have to keep exactly the same to make sure that the one thing you're changing, the temperature in this case, is actually what's causing the results you see. And wow, the mark scheme gives you a ton of options here. You could have said the volume of milk. Makes sense, right? More milk might take longer. Or exposure to air or oxygen. You could talk about sterilizing the test tubes, which is super important so you don't have rogue microbes messing up your experiment. Even the age of the milk or its starting pH would get you a mark. You only needed 2 from this list for the full 2 marks. Lots to choose from. Okay, so we've covered the setup. Now it's time for the fun part, looking at the data. The next few questions are all about making sense of the results, and that means we need to get friendly with a graph. So let's just take a second to break this graph down. Along the bottom on the x-axis, we've got temperature, and up the side on the y-axis is the time in days for the milk to hit pH 5. And you can see from the key we're comparing 3 different types: cow's milk, goat's milk, and almond milk. Got it? Cool.
Okay, first question, using this graph. Which type of milk stays fresh the longest at 10 degrees Celsius? This is a straightforward data pull. Find 10 degrees on the bottom axis and just look up. Which line is the highest? The highest line means it took the longest to decay. And yeah, you can see it right there. The dashed line for almond milk is way up top, so almond mark is our answer.
All right, a 2-marker now. Describe the effect of temperature on the time taken for goat's milk to reach pH 5. The key words here are describe and use data. Though, interestingly, the mark scheme here doesn't actually need you to quote specific numbers. Our job is to focus only on the goat's milk line and explain the pattern as the temperature goes up.
So how do you get both marks? You have to spot that the line has 2 different parts. The first mark comes from describing the first part. As the temperature goes up to 15 degrees, the time it takes to decay decreases. Simple enough. The second mark? That's for spotting the change. You need to say that above 15 degrees, the time taken to decay pretty much stays the same. It flattens out. Describing both parts of that trend is how you lock in those 2 marks.
Okay, we've described what the graph shows. Now it's time for the big question, why? This is where we need to dig into the science and explain the biology behind those patterns.
So, we've got a couple of why questions. First, why does milk decay faster at 10 degrees compared to 5? Well, it all comes back to those microorganisms. Think about it. When it's warmer, they've got more kinetic energy, their enzymes work more efficiently, and they can divide and multiply much faster. Any one of those reasons would get you the mark. And the second question, why do the different milks decay at different rates anyway? This comes down to what's in the milk. Maybe they have different amounts of fat or lipids, or perhaps they start with different types of bacteria already in them. Even a different starting pH could be the reason.
All right, we're on the home stretch, the final part of the question. This is where they ask you to think like a scientist and figure out what the next logical step would be for the investigation.
So the question poses a new idea, testing the link between storage temperature and the risk of food poisoning. What's the best next step? You get 3 choices. A. Determine the types of bacteria present in the milk. B. Record the pH every 12 hours. Or C. Use more than 3 different types of milk. Which one do you think it is?
The correct answer is A. Determine the types of bacteria present in the milk. And here's why that's the only real answer. Food poisoning isn't just about milk going off. It's caused by specific, nasty, pathogenic bacteria. Just because the pH has dropped doesn't mean the milk is dangerous in that way. To actually test the risk of food poisoning, you absolutely have to know which bacteria are growing in there. And there you have it. That's how you think like an examiner, break down a question, and make sure you're grabbing every single mark you deserve.